Test test 1 2 3. Hello 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 hello. Test test. Mic check mic check. Hello 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 hello. Mic check mic check. Testing 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 1 2 3. Testing testing.
Hello. Oh, yeah, this is working. Hey, everybody. We're going to start in about 2 or 3 minutes. So just so that you're aware, about 2 or 3 minutes, what will happen is the trailer will start and that'll happen. And then I'll talk again because this is what I do. Toodaloo.
Close to somebody's heart, it's Nerd Night. Get your nerd on in Tokyo. Keep your shirt on. Yozen, we'll make you think now. Park Lounge, buy a drink now. We're here, and it's time to begin. Pay your money to Selena and get yourself in to a chair and maybe a drink. Tonight will really make you think. It's Nerd Night. Get your nerd on in Tokyo. Keep your shirt on. Yozen, we'll make you think now. Park Lounge, buy a drink now. Time to sound off. It's Bruce. He arranges the talks. He doesn't drive a car. He just loves long walks. It's Melvin. He films the live stream. He also loves long walks and eating ice cream. It's Coco, making sure things stay on track. When all the shit goes down, she's always got our backs. It's Selena, she's quite a find. The friendliest person to ever rob you blind. Give her your money now, it's Doria, she runs the sound. We couldn't hear Jack if she wasn't around. It's Andrew, just take a glance. I don't think he actually owns any pants. It's Nerd Night. Get your nerd on in Tokyo. Keep your shirt on. Lowe's on. We'll make you think now. Park Lounge. Buy a drink. It's Nerd Night. Get your nerd on. Keep your shirt on. Lowe's on. We'll make you think now. Park Lounge. Buy a drink now. Here we go.
Is it? Oh, okay. Hi, welcome. It's been a heck of a setup today. Everything. It's basically Murphy's Law Day here at Nerd Night number 65. Titled Digital Biology, which I'm told is not about finger stuff. Tonight's... That's the best joke I've got tonight. I'm telling you, it's been a rough setup. Tonight's music and trailer song, again, by Algorithm. I wrote the lyrics, but yeah, Suno.com wrote the thing. And actually, hold on, I've indented stuff so much I can't read it. It's one character wide. Let me just rotate the screen. Oh, time is we live in. Daria is no longer Daria, but now she's Doria, according to the algorithm. And last month we had trouble with the word Sugamo, or last in December we had trouble with the word Sugamo, and the singer would say Shugamo. So I tried writing Ryozan in katakana, but I think what happened is it transliterated it to Romaji and then read it with an American accent because it's not close. It still couldn't get it right. Anyway, Nerd Night, welcome. It's the local franchise of a global speaking event. There are Nerd Nights all over the world. There are 4 Nerd Nights in Japan alone. Every month, we host 2 speakers who talk about their field of expertise, or not. So these can be science communication talks, also talks about arts, humanities, activism, weird hobbies, digital biology. Help us out by following us on social media. We're mostly on... Actually, we're not mostly on anything. It's up to Bruce and I to do the social media posts. We both have better things to do, apparently. But when we do post, we are on Mastodon and still on Facebook by necessity. But we do not post any longer on the platform of the asshole. My public service message, please use Firefox, blah, 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 blah. But even though Firefox is a little bit crappy, it's still better than the alternatives because it's technically a nonprofit. Okay, let's go to the next card. So can someone hit spacebar on my big computer there? No, that was it. Oh, wait, hold on. You had to be on the right screen. Just a second. Try not to get feedback on the mic. No, it's on the, you're on the, sorry, there we go. There we go. Okay, so our next event is on March 21st. And just pay attention. We are not here on March 21st. We're at Blink in Roppongi. Because we like getting harassed by touts, apparently. Yeah. So, and it's March 21st, which is Nasty White Day. No, it's just me. If you were here in December... This is a continuation of a joke about Nasty Valentine's Day. But anyway, okay. Yeah. And here we are. But this month, we're not there. We're here in Ryozan Park Lounge, where you go in Sugamo for Golden Years speed dating. By the way, if you want to speak at a future event, don't talk to me. Talk to Bruce. Bruce, put up your hand. He's the man with the mask at the back by the mirror. Talk to him. Right, next card. Beakers. We only have one. So if you want to buy a beaker, we only got one tonight. My bag was too full. I couldn't carry any others. So, yeah, that's 1,500 yen. We're not going to demo it because it's just one. But it looks like that, and they're cool. And, yeah, next slide. Call for volunteers. Oh, my God, the setup today. This would be so much easier if I only had one job. And the way that would work is if someone wanted to volunteer and, I don't know, take over... running the video setup, maybe the audio setup, maybe the stream setup. That would be great. Or MCing. We lost our temporary MC, who was, or not temporary. The guy was I was supposed to be temporary. He's off working on the expo or something. Ren's away this month. So yeah, it would be great if I only had like 1 or 2 jobs per show. So all these things, we need those. Next slide. Hey, let's be cool and join the mailing list. If you're not on the mailing list, you can use that QR code to join it. I think we've also got, do we have it somewhere else? It's up the front. There's a QR code or a sign-up sheet or something. Okay, QR code up there, and we can always take your email address by paper if we still do that. I used to make dick pic jokes here, but I'm reformed. So next slide, please. He's still there from December. Noel, get out of here. It's ongoing. That was my, yeah. I used to say just we only send tasteful dick pics, and then I would have a picture of someone named Dick. That was the joke. I told you I don't have anything good tonight. Okay, here's the agenda. Tonight, I've got Joel. Oh, you know what? I did not. Joel. Is it Nitta? Yeah. Okay, it's not. Okay. I assume, but it's good to ask. First, Joel Nitta will explain why ferns are so fascinating. Hopefully he's not talking about my Aunt Fern. Because that would be weird. And she is very interesting, though. She's like 86 and climbs the Grand Canyon and crap like that and vaults over the Grand Canyon or whatever. Anyway, so he's going to talk about why ferns are so fascinating and how he's been using methods adapted from open source software and data science to better understand their ecology and evolution. Our second speaker is Louis. Oh, Jesus. I also didn't have time to ask you, is it Kang or Kong?
Kang.
Kang, okay. So like Kodos and Kang. Exactly, okay. Yeah, he's going to give us a guided tour of the neural network research that led to the 2024 Nobel Prize in Physics, which I'm sure we're all very familiar with. Work that gave us insight into both biological and artificial brains and underpins today's artificial intelligence. I don't know what any of that means. You know what? Maybe I need a neural network to figure that out. I told you I had no jokes tonight. After each talk, there will be a Q&A. Please wait for the mic to ask your questions. Try to make it a question, not a dissertation. I know a lot of us here like to make dissertations, but let's not do that. Okay, let's go to the next slide in case I forgot something, but I think we're on to our first speaker unless oh, no, I'm promoting this. There's a dude whose name isn't on here, so I can't remember. Do you remember his name? Michael something. Anyway, he is doing interactive video game concert on February 28th at Blink Roppongi, which is the place where we're doing our event in March. And it just didn't work. We were thinking maybe he'd be at a Nerd Night, but timing-wise, it just didn't work out. But we told him we'd let you know where it is. There's a QR code. Yeah, you could probably just look up Extra Lives. Maybe, I don't know. All right, next slide. Now we should be there we are, Joel Nitta. Okay, now I got to introduce you again, Joel. That's just how it works. That's just how it works, man. Joel is an associate professor in the Graduate School of Global and Transdisciplinary Studies at Chiba University, Japan. He studies ferns, an ancient and diverse lineage of vascular plants. This is literally all he gave me. Oh, wait, sorry. He is also, and I quote, also an enthusiastic adherent of reproducible analysis and coding, especially with R. Did that mean anything to anyone other than... Okay, good. I think there's a pirate joke in there somewhere, but I couldn't get there. Pirate Ferns? I give up. It's been a long week. All right. Joel, why don't you come up? And yeah, I'll switch over the video stuff. I'm going to leave this here in case your microphone fails. Oh, I forgot it. This is hard to do with one hand. There's going to be mute my mic because there's going to be a loud noise when I plunk it in. I will stop you rather than allowing it to everyone don't put their hands on their ears. So
okay.
I'm running up. That's what's going on.
Okay.
Anyway, put your hands together for Joel Nitta. Let me just switch this over. Wait a second. Fingers crossed, everybody. Nope.
Nope. Okay.
And let's hope the clicker works.
Okay. Hi, everybody. Thank you very much for having me tonight at my first Nerd Night. I'm very honored to be able to give this talk for you all about open source ferns. So yeah, a brief reintroduction of myself. My name is Joel. I'm a fourth generation Japanese American, originally from California, now living here, professor at Chiba University. And my research topic is the ecology and evolution of ferns. Now, when I was putting together my slides for my talk tonight, I had to answer kind of a surprisingly difficult question. Why ferns? You know, when you study something for long enough, you can kind of get lost in the details. You know, you can lose sight of the forest for the trees or the fronds in this case. And, I could give you sort of like some stock answers, like, ferns are important for biodiversity, which is one of the Sustainable Development Goals, or that, fern-like plants form most of the fossils that are now our fossil fuels. And, those are all true, and they are important reasons, but they're not, when I think about it, that's not the real reason that I got started studying ferns. I think the reason that I study ferns is that ferns are mysterious. And that, it doesn't sound very scientific maybe, but it's true. So ferns, they lack flowers. They all kind of have this uniform greenish appearance to them. But, beneath that, it turns out that they're actually quite diverse, and they have a lot of really interesting aspects of their biology, which is don't. Okay, I won't. All right, sorry. Excuse me. So they're just very cool plants to study and find things and make discoveries about. And I first experienced this as an undergraduate at UC Berkeley that has a special field course on the island of Moorea near Tahiti. If you ever have the chance to take a course on tropical biology in Moorea, I highly recommend it. It was a beautiful island, but even, the bigger reason is that during this course, for our final project, each student had to choose a particular organism on the island and study that. And we're there for almost the whole semester, right? And a professor happened to recommend that I look into these ferns. And so it's this group of ferns called the filmy ferns, or the family Hymenophyllaceae. And they're unlike any fern I'd ever seen before, because they only grow in very damp, mostly in tropical areas. We do have some in Japan. I'd never seen any in California. And they're unlike any fern I had seen before. They're really, really small, and they tend to have very, very thin leaves. They're almost translucent. And so when I was able to spend a lot of time looking for these things on this island, I became very familiar with them. And you kind of get a sense for not only what they look like, but the habitat where you might expect to find it. And so I was able to find this particular species that had never been recorded at least in written histories from the island before. And in collaboration with some local botanists, we were able to publish that. And so being able to make a discovery like this as an undergrad really just hooked me on ferns, and so that's why I've been studying them ever since. And you can see, like, if you saw this thing out growing on a rock, you would probably just think it was moss, right? But if you look up really closely, it is a fern, and it's quite small, like less than a centimeter in size. Now, I'm not the first person to kind of have this sense of ferns as being sort of mysterious organisms. Actually, for a long time, ferns were grouped together with some other things here, like moss and even some mushrooms, yes, and seaweed and such. Thank you. English wasn't coming to me for a moment there. As a group of organisms called the cryptogams. So crypto, meaning like, mysterious or hidden, right? And the reason for that is, what do these all have in common? Well, it's what they lack. They don't have flowers or seeds. So at that time, at least people in Europe couldn't figure out how these could possibly reproduce. They were these just kind of weird, mysterious organisms that all got lumped together as the cryptogams. And in fact, there's a quote from Shakespeare, Henry IV, "We have the receipt of fern seed. We walk invisible." And the idea was that ferns lacked any obvious seeds or flowers, so, and yet they reproduce. So that means their seeds must be invisible, which means if we have the fern seed, we are invisible too. Of course, now we know things don't work like that. But so the point is that, yes, ferns are a thing of mystery. And so when we think of ferns, what do you picture? Well, most folks probably picture something kind of like this, a fairly bushy plant growing in the shady forest understory. And that is, true in part. But I would like to point out that that is not all there is to ferns. they're actually much, much more diverse than that once we start to scratch beneath that surface. There are ferns, there's many fern species that grow, in fact, in the desert in extremely inhospitable situations, like on this rock here. There are those, again, the really thin ones that I was telling you about that I like. Those are the filmy ferns, and you can almost see, that leaf is like partially translucent. And then, of course, there's ferns that grow as big as trees, the tree ferns. These are some that I saw in New Zealand recently. And then we even have aquatic ferns. And those are the things that look like four-leaf clovers growing in the water. And ferns grow on every continent except Antarctica and in all sorts of different habitats. So yeah, they are actually quite diverse. But what ties all of these diverse things together? Well, as I sort of alluded to earlier, they don't have flowers. Instead, they have spores. So that's one thing. They've got spores. And they also have what we call vascular tissue, which is if you, cut the stem like this, you would see these little dots, which are actually pipes running long ways, and that moves water through the plant, right? So those are two pretty easy characteristics, I think, that you can use to identify ferns. But here I should point out something, and that is that actually, ferns are not the only land plants that have those characteristics. In fact, there's another group. So here we've got ferns. There's another group right over here, which are, is this also a pointer? No. Okay.
Yes,
no.
Not that one. Oh, the other one.
It's all right. Called the lycophytes. And so historically, these had been grouped together into a group that you may or may not have heard of called the pteridophytes. And I put quotes here because we now know, thanks to DNA sequencing, that in fact the ferns and the lycophytes are not actually that closely related to each other. They just happen to have these very striking characteristics of vascular tissue and spores. And this sort of diagram, I'm going to explain a little bit more detail because it's going to come up again later. We call this a phylogeny, or an evolutionary tree. And so these lines, those show the connections between things, these organisms at the tip, and how they're related to each other. And you'll notice that, if the ferns and lycophytes were closely related, that those lines would come together there, but they don't. We have over here the gymnosperms, so that's things like pine trees, and the angiosperms are, plants with flowers, and those are, grouped together. And then actually the ferns are more closely related to them than they are to these guys over here, the lycophytes. And this may seem like a kind of, a technicality, but this was like mind-blowing for botanists when we figured this out. And again, that was only thanks to DNA, because none of this is apparent from what they look like. So that is, one example of how these sort of modern techniques are uncovering some of these mysteries of these plants. Now, I'd like to go back to our friend, the fern in the forest here, to show another what I think is a fascinating thing about these plants. And that's it. If you were to look a little bit more closely here at the base of that fern, you might see something like this. And if I would ask you what that is, you might not know, but I'll give away the answer. This is also a fern. And that is the tip of a mechanical pencil there for scale. So you can see how small this guy is. And that's its full size. It doesn't get any bigger. And these are actually the two halves of the fern life cycle. Now, again, I don't want to go into too much details here, but we can see how they're related through this life cycle, which is in every biology textbook. On the left, and it's these two halves, and the technical names are the sporophyte and the gametophyte. And the sporophyte is what we typically think of as a fern. And sporo is for spore, and phyte is for plant. It's the spore-bearing plant. The spores are released into the wind, they land somewhere, they start growing, and they don't immediately grow into another ferny-looking thing. Actually, they go into that little guy that I showed you earlier, and that is called the gametophyte. And, oh, that's a bit cut off, but there, gameto is for gamete, or eggs and sperm. So the plant that produces the eggs and sperm. And that is where the sex happens, and then we have a new sporophyte coming out of the gametophyte. And so this is, to me, another great example of how ferns are mysterious, because when I go out and I walk in the forest, I know that if I look carefully enough, I can find these little hidden fern gametophytes in sort of like, it's kind of hard to describe, but in if you know in the right places to look, which are oftentimes little kind of nooks and crannies almost amongst the rocks and roots and things like that. And the thing about these gametophytes, though, is that most of them, this is very representative, they almost all look like that, just little heart shape. And in fact, a trained botanist, although I might be able to tell you that is a fern gametophyte, I don't know what species it is. I could not tell you just from the way it looks, because many different species look just like that. And so for that reason, for many years, even though gametophytes are the site of fern sex, and therefore really important in the fern life cycle, we just didn't know much about their ecology, because it's hard to find them in the first place, and once you do, you don't know what species you have. But recently, that has been changing, again, thanks to DNA sequencing, right? So this is a technique often referred to as DNA barcoding, where in order to identify this, what we call a cryptic life stage, because we don't know what it is based on its appearance, you can sequence the DNA, query that against a database, and get a match and figure out what the species is. And so I've been using that in my research to uncover things about fern ecology that were previously unknown. And so this actually is where we finally make the connection to open source. And you may be like, wait a minute, what's this open source have to do with fern gametophytes? Well, the important part is this part over here on the right, because I can go out and collect fern gametophytes and sequence their DNA in my lab. That's fairly straightforward now. But the critical part is having the reference database, being able to query that and get a match, right? And so fortunately, there is a really important database that is, it is run by or supported by the US government called GenBank, where almost every researcher deposits, whenever we publish a study that has DNA records in it, we put a copy of it into GenBank, and that is made open. There's usually a short embargo period before your paper gets published, but once it's published, the data become open, and anybody can use it. And so that is hugely important as a biologist, because without that, it's going to be much harder to really say anything about the DNA that I sequenced in my organism, right? And so I think you can see here how, if we compare this, to open source software, for example, the famous open source operating system Linux, which pretty much forms the backbone of the internet and many mission critical databases. It's even on probes sent into space. We will make huge progress thanks to open source software. Just in the same way, open data are really critical enabling scientific progress. And so with that in mind, for the remainder of the presentation, I'd like to give a few examples of how I've been sort of taking this open approach to my own research on ferns. And so the first of those is called the Fern Tree of Life, or FTOL project. The goal here is, well, fairly simple, to build a phylogenetic tree, that was that diagram I showed earlier, including all the fern species on the planet, which is about 12,000 total. And we have a website. Unfortunately, that's tiny, but if you Googled Fern Tree of Life, you would probably find it. And so GenBank comes into play here again, because in this case, I'm not using it to identify ferns. I'm analyzing that data directly and using it to figure out how different species are related to each other. And so when I was working on this analysis, I used code to automate the whole process. And as Andrew mentioned in his introduction, my favorite programming language is R, and so I used R to automate all of this. And the beauty of that is that this database is constantly growing as researchers publish new papers and put out and add more DNA sequences to it. And so any tree that I build today is going to be out of date quite soon. And so in order to make a phylogenetic tree that reflects our most up-to-date understanding of these relationships, I need to keep repeating this on a regular basis. And so as a result of doing that, the initial publication of this fern tree of life, happy to say, it was the largest fern phylogeny to date, includes about 6,000 species, so we're around halfway there. And every time I rerun the code, which is, well, depending how busy I am, around every 3 to 4 months, something like that, then we have a few hundred more species, coming in. And so it's been steadily increasing since the initial publication. And that was in 2022. And so, I mean, this was, I was very happy to get this out there, but what I'm even more happy about is the fact that other researchers are using this as a resource. In fact, it's been cited over 50 times since it was published. And here I'd like to show an example of what I think is a super cool study that used the fern tree of life. And they were looking into this relationship that ferns have with ants. And in fact, it turns out that some ferns produce nectar. I didn't know hardly anything. I didn't know anything about this before I saw this paper. And it's a really, I think it's a really fascinating study. So it turns out that although we normally think of nectar as something that flowers make to attract pollinators, it turns out that some other plants do it as well. The ferns do it. And you can see here we've got this little nectar drop produced on something that is definitely not a flower. And the reason they do that is they have a symbiotic relationship with ants that feed on the nectar and then protect the fern from other insects that might try to eat it. And you can see the tree here is, that's from my study, the fern tree of life. And what they've done is the spots, it's a little bit hard to see, but they have spots in, you can see these little spots in green and then that are connected by the green lines. And so those are groups where we have this, what we call a trait, right? This nectar production and the relationship with ants. And they were able to date the tree, this is we have millions of years ago, and figure out that they think that this trait evolved around the same time that it evolved in flowering plants, because they analyzed it in flowering plants as well. And they think it has to do with ferns that grow as epiphytes on trees, that when they moved into the trees, they picked up the ants from the trees. So I just think that's really cool, and I'm very happy to see that the fern tree of life is able to use this way, and that this is a great example of how that I'm able to build a tree based on open data and then put it out there, and then other researchers can then build off of that. All right. Now I'd like to change the focus a little bit to this other project, which is about taxonomy. And taxonomy is the study of naming life. How do we put names on species, basically? For example, we are Homo sapiens, genus Homo, species sapiens, right? And this is a system that was developed by Linnaeus back in the 1700s. And it's actually kind of amazing that it's been used in more or less the same form all but ever since that time. And another thing that has been that has continued for a long time is choosing which names to use. So Linnaeus described the set of rules for how you name things, but then actually naming things is a different matter. And then deciding which names to actually recognize is also a different matter. And so for a long time, that had typically been done by a small number of people, often white men in Europe, and they would publish a taxonomy that is based on their assessment of the field at the time. And so there's a couple examples in ferns. But as we can see from those previous slides, we are sort of awash in data these days about DNA data, and there's constant activity going on and publications of new relationships and new species and such. And so this model isn't maybe the best, is not maybe the efficient or the best way to go anymore. And so recently, as a community, researchers on ferns and lycophytes have gotten together to try to come up with a new way of doing this sort of taxonomic work. And we call this the Pteridophyte Phylogeny Group, or PPG. Remember, pteridophytes are ferns and lycophytes. And this is a picture of us at the International Botanical Congress in Madrid last year. Our goal is to democratize and accelerate the taxonomic process by using an open source, community-driven model. And I'm quite happy to say that our group is large and diverse. We have, this is only the people who are in person at that particular meeting, but we actually have close to like 200 members from nearly 60 countries, all different ferns and researchers on ferns and lycophytes. And so this is an example of how we carry out our discussions, because of course we're all over the world, right? So we, so before I got involved, they used to do it using email. But email can get a bit unwieldy, and it's also not open, right? It's only available to whoever's on that email chain. So we've recently switched to hosting discussions on GitHub, on a public repository. So all of, and what we do is, so if you've heard of GitHub, you probably are familiar with it in the context of software and software development. But GitHub, and as part of that, GitHub has what they call the issue tracker, which is where you can submit bug reports and then have discussions about them. And it turns out it actually works really well as a public bulletin board. So that's what we've been using. And we have one issue for each taxonomic topic that we're talking about, such as a new genus and whether we should recognize it or not. And I think maybe I should explain a little bit more what I mean by that, is that we are not publishing new species and new names. Someone else does that. But then the thing is, scientists don't necessarily agree on the way to carve things up. Like, you could split a genus into two smaller genera, or you could recognize them all in one single genus. And it's not always clear. And there could be people who've published this paper that does this, and people who publish a paper that does the other way. And so it's not necessarily clear which is the best approach. And so that's why we're taking this democratic way to do that. And so we carry out our discussions here, and those are all available online for anybody to see. And then we have a vote, which is actually using Google Forms. I take the result of that, and then I post it back on the GitHub so we know what the decision was. And so by doing that, we've worked our way through 80 genera that were recently published, and I'm very happy to say that in collaboration with the group called World Flora Online, our system is now available for anybody to use. So if you want to know, does PPG recognize this genus or not, you can go to this website and have a look there. I mean, it's mostly for researchers, but anybody can, of course, use it. So yeah, that is what I wanted to tell you all about. And so I would just like to wrap up with a few take-home messages. First, and most importantly, is that science thrives in the open. In academic research, you've probably heard of this cliche phrase, publish or perish. And unfortunately, it's true. That is still, publications are still the primary currency in academic research. And so for that reason, there's been this tendency for researchers to hold on to their data because you don't want to get scooped. But I've found that is almost never the case. I think if you make your data open as early as possible, it enables collaboration. Other people can get involved. And there's way more than any one of us can possibly do. So helping each other out is really important. Another thing is that we need to incentivize the maintenance of databases and software. So like I said, again, there's still this unfortunate truth that publications are king in academic research, but we can't make new findings and new publications without up-to-date data, right? And so I find it very important it's very important to me and to support my fellow researchers to have this up-to-date fern tree of life. So I put a certain amount of time into that to make sure that it's up to date. But that's not actually a new publication that's going on my CV, right? So I think the system needs to change to recognize how important that is. And finally, ferns are cool.
Oh, okay. Are we on? Okay. All right. So now it's time for the. I'm not gonna get up. Sorry, too lazy. Now it's time for the Q&A. So if you have questions, not dissertations, raise your. Catherine, right in the front. Here. Oh, sorry, Catherine. I thought that was your. All right.
I was like, what happened? Did it look like your question? So it's not. I guess it is kind of a dissertation, not a question. Yeah, I think I can hear myself. It's on GitHub. You can put your org ID in, and then right now it doesn't, I think, count for anything. But I think the plan is eventually your activity in GitHub will start showing up on org ID.
Oh, really?
Yeah.
Cool. Oh, thanks for letting me know. I'll have to check that out. That'd be great.
Question.
At one point, you said we are not the one who is going to decide what the taxonomy is, but there is a discussion and having a popular vote to see, okay, so who actually makes the decisions in taxonomy, and what are you doing to make sure those popular votes and discussions have any power in the ultimate decision?
Okay, so, all right, so the way it works is any researcher who follows the rules of taxonomy can publish a new name, for example, a new species or a new genus, because as long as they follow the official rules. But the thing is, people can have sort of conflicting interpretations. There could be two different names, and you could recognize one or the other, but not both, right? And so what we're doing is establishing guidelines for ferns and lycophytes of the way to use these names that have been published that we think makes the most sense. And then as far as anybody follows those guidelines, that's up to you. It's not legally binding or anything. This is just the consensus opinion of 200 pteridologists. Yeah. And in fact, there is another prominent website with their own global treatment that differs from ours, and we can't tell you, which one you. Well, I can tell you which one I think you should use, but it's up to you. Yeah.
Thank you for your. Oh, that's loud. Thanks for your talk. I'm a PhD student, and we get told all the time about the importance of open data. So this is really cool, and I might take it back to my
Please do.
University.
Yes.
Because they love good examples. But you mentioned the NIH databases from America. Obviously, with what's going on there, I was just wondering if you know what's going to happen to them. Are they going to keep being there? I really hope so.
Yeah. I mean, I was just on GenBank yesterday. I mean, I'm on it pretty much every day. And it wasn't loading. And then I'm like, what's going on here? And there's a little banner at the top that says, we are experiencing heavier than normal traffic. I don't know what that means. But fortunately, at least with GenBank, there are global mirrors, copies of it, one in Japan and one in Europe, the DDBJ and the EMBL. So at least we have that. And that is another really important thing about open data, is that it can become distributed. Right? It doesn't have to live in just one spot. So yeah, that's really important. And I hope to God that somebody doesn't get his hands on that database in the States.
Thank you for the presentations. I want to ask a little bit about fern. So evolutionary speaking, is there any advantage of having such a peculiar reproduction method, let's say?
So yeah, maybe about the sporophyte and the gametophyte. So it's hard to say. It would be really hard to devise an experiment to test for an evolutionary advantage or a selective advantage to that particular life cycle. But we do have some ideas. For example, because those gametophytes are so small, they can grow in places where the sporophytes cannot tolerate. For example, they can grow in sort of these, like if this table, there's a little like a nook here, it could grow in that nook. And if this table was in the desert, that nook could be actually really well protected and not get so dried out. And so by doing that, the gametophyte can survive there even though a sporophyte would not be able to, because it's so big, it would not be able to survive. It could not hide out and be protected from desiccation. And in addition, some gametophytes, in addition to being small and being able to in those nooks and crannies, some of them can just flat out handle being dried out, and the sporophytes can't. So that gives them some sort of flexibility in where they can grow. So I would interpret that as possibly an advantage, but whether that really helped them survive over evolutionary time or not is an open question. Yeah.
Thanks for the talk, Joel. Have you named a fern? I'm guessing you have. And why did you name it what you named it?
Oh, thanks so much. I have been fortunate enough to name a fern. It was during my PhD research, so for my PhD research, I went back to that island because it's a beautiful island. And I got to know the ferns fairly well there. And so I was collecting ferns on the biggest mountain of the island because as you go up in elevation, the species change, right? So, and I noticed this one that looked, I couldn't match it to anything, and it couldn't match it to anything else that was described from there. And so it turns out that it's actually a hybrid between two fern species. And so I named the hybrid Microsorum tohieiense. And tohieiense is after the mountain where I collected it, Mount Tohiea. Ense means of this place. Yeah.
Thanks.
Thank you also for this interesting talk. I'm just wondering whether the study on ferns is only scientific, or does the ferns have any aim in the biological life chain? Like, for instance, are they necessary for human life or anything else like this?
Okay. I would say they're necessary for enjoyable human life. I mean, ferns have, I believe there is some research into medicinal usages of ferns, and they're used ethnobotanically in certain cultures. For example, ferns produce a number of chemicals that deter insects from eating them. And so, yeah, some of those have been the targets of studies for medicine. I don't know how much has actually, come out of that, but yes. And yeah, and they are, of course, quite nice to enjoy. And if you're interested in getting to know more about ferns in this area, there's a group called the Japan Fernists Club, I think it is in English, or Shiranokai. They meet monthly, and anyone is welcome to join and go on outings and learn about ferns. Yeah. And they're amazingly knowledgeable. I learn every time I go with them. Yeah.
Thank you for the talk. So just to clarify, the gametophytes don't spiral up.
Don't what?
Spiral up, do they?
I'm sorry, I don't.
The gametophytes, they don't curl.
Oh, curl
like
up.
that.
Okay,
Yeah.
so yeah, the uncurling of the frond, that's only in the sporophyte.
Is there a reason why?
I think it would be developmental because of the way the fern frond is shaped. Gametophytes it's not a frond at all. It's just completely different. It's a, well, sorry, I don't know about the earliest, like the very earliest few cell divisions, but once you get to anything resembling a leaf, they're completely different. So yeah, it does not do the unfurling.
Okay, thank you.
Yeah.
Just a quick announcement before we take a break. We have a few open seats around. There's actually a very comfortable lounge up here in the front where you get a great view of the speakers and you can kind of just relax and lay back. There's a few seats that might be open along this back wall here as well. So as you get up and rearrange during the break and everything, please try to help people find a seat and enjoy the break.
And sorry, I forgot to actually read the acknowledgments, so if I may. I do want to acknowledge everyone this is a huge team effort, especially PPG. And so I have the names of some of the other PPG committee members there, and especially Michael Hassler actually came up with the first version of the data that we use, so that's huge kudos to him. And then also the JSPS for supporting my research. And thank you all for listening.
Whoa, I'm gonna fall. Everyone be careful when walking around here. There's lots of cables, more than usual. Joel, thank you so much. We're gonna take about, is this even? Hello? We're gonna take about a 20-minute break now, and then we're gonna come back with our second speaker. So, if you haven't got a seat, come in, find a seat. And once more, a big hand for Joel. Thank you so much.
Thank you.
Keep your hands off my aunt.
little person, little jumpsuit wearing sequins. Well, it's time to stop your midnight driving and put your dancing shoes away. A little woman, little man, little grown up, little platform wearing disco fan. It's time to stop your late night shaking. It's the end of a long dancing day. This is a disco. This is a disco lullaby. Lay down your funky head. It's time to go to sleep now. Go get tucked into bed. Your daddy and your mommy will boogie shake their bunnies at the disco scene. But first they'll tuck you into bed and you'll disco in your dreams. Disco dreams. Oh, little boy, little girl, little person, little jumpsuit wearing sequins. Well, it's time to stop your midnight driving and put your dancing shoes away. A little woman, little man, little grown up, little platform wearing disco fan. It's time to stop your late night shaking. It's the end of a long dancing day. This is a disco. This is a disco lullaby. Lay down your funky head. It's time to go to sleep now. Go get tucked into bed. Your daddy and your mommy will boogie shake their bunnies at the disco scene. But first they'll tuck you into bed and you'll disco in your dreams. Disco dreams.
dreams.
Just so everyone knows, we're going to start again in about 5 minutes, but then I'm going to talk a bit. So, like 7 minutes, 8 minutes, 9 minutes. But just, heads up, if you need to get a drink, do it now or while I'm talking. That's fine too. Cheerio. How do you say goodbye? What's the protocol? Wow. Sorry, whoever was right by that speaker. All right, we're back. So this is where I'm supposed to tell you to. This is where I'm supposed to tell you to buy beakers, but did we? Bruce. What's that? Don will buy the beaker. There's only one beaker. Don wants it. Did Don get it? Don, did you get the beaker?
Okay, everyone, if you would like an incredible, if you would like an incredible, one of a kind, priceless.
It's one of a kind tonight.
Yeah, one of a kind tonight. We have it. But 1,500 yen, it is yours. But if not, it's going to actually win Don. Please raise your
Yes.
hand now. Going once, going twice, okay.
Oh. That arm wrestling is a normal human thing to do that Don Warren Human will do. I'm misintroducing you, Don. Don used to be a member of the Nerd Night team. He used to sit on do this sort of slide stuff, so I wouldn't have to keep messing with it like I have had tonight. I miss you, Don Warren Human. He's totally a human. Okay. That's what they say. Okay, where are we? Can someone hit the space bar for me, please? What have we got up here? Oh, yeah, our friends, Tokyo Humanities. Is anyone here from Tokyo Humanities Cafe tonight? Because I don't know when their next event is. The QR code is there. You can go there, and if they have another event coming up, I think you'll be able to see it. I'll hold that on the screen long enough. I don't see a lot of phones coming out. I see 1, 2, 3 phones. Okay. All right, you're just typing. Okay, you got it. All right, yeah, let's go to the next slide. I think it's Space Cafe. They also have not told me when their next event is. I don't think they've booked it yet because I looked at the website and I had the event for. Oh, we're jumping. Oh, it just went by itself, did it? So, okay, now I got to slide ahead and. Okay. Right, Nerd Night Tokyo is produced by the Yokohama Theater Group, or YTG, which are the initials you see on the screen there. YTG is a registered nonprofit and is the beneficiary of any net income Nerd Night is able to produce. From the sweat of my brow, working on an audio project. So we're working on an audio project set during the Beatles' tour to Tokyo in 1966. That's going to be released for free as a podcast, probably sometime in 2026. It's a doozy. We've started work on our next show, a very strange piece called Interference. It's going up. We're doing a showcase, which will be a work in progress showing at the beginning of April. So I'll announce the details at the next Nerd Night, but that'll be April 5th and 6th for sure. It's very weird. It's very strange. Yes, it's incredibly strange. Anyway, your money, I guess I was trying to say something fancier than that. Your money it goes to support the company in making the projects I just discussed and many others. And because it's impossible without, I'm popping my peas there. It's impossible without ongoing support to produce live international theater in Japan. Okay, let's go to the next slide because I feel like I missed something fading. Oh, no, I'm just going to introduce the next speaker. Louis Kang. Louis Kang is a principal investigator at Riken Center for Brain. Okay, I made it sound like he was going to be like a detective or something. He's a principal investigator at Riken Center for Brain Science, working in computational neuroscience. Don't have any jokes about that, sorry. I did not get a lot of biographical information from him. That was something I could repeat here. Not because of profanity or anything. Just it was a lot of like listings of degrees and stuff. So he could tell you all about that. But I had to do a bit of cyber stalking to find some actual like sort of biographical information. So apparently he likes to play piano and beach volleyball. Is there a joke there? Does he do them at the same time? I know that's kind of obvious, isn't it? The same. Anyway, I asked ChatGPT to give me some info on his hobbies and his personal life, and it told me rather unhelpfully, he probably enjoys intellectually stimulating leisure activities. Thanks, ChatGPT. Thank you. ChatGPT has gotten a lot less creative recently. It used to make up really weird things, and now it plays it safe. Anyway, please give a warm welcome to Louis Kang while I walk over and press the button that'll turn the screen, bring up his Don't cut me off mid-sentence, god damn it. Yes, big round of applause for him, and just keep that going until I get this switched over.
Awesome. I'm Louis, thanks.
Oh.
Give
it a second.
Just need I feel like it needs a drink. Maybe a little.
This is literally the first time this ever done this. This is new.
We can try it to see.
Hold on, let me just. You're not supposed to do that, by the way. Okay. That's not for the. Okay.
Oh.
Okay. This is just the way tonight's gonna go, isn't it? So,
exit the presentation and try again.
I don't think the presentation is the problem. If it was.
Do you have any like secret scatting skills or dance skills?
Oh. Okay. It's true. I can do a little river dance. Okay. It was even the threat of me scatting immediately, shocked this presentation to working. Exactly, yeah. Thanks for having me here. I'm Louis, and yeah, I'm going to tell you a little bit about neural networks today. Perfect. So the most remarkable feats of intelligence that we know today are performed by networks of neurons. So in both biological brains and also in machine learning networks, there are millions upon millions of neurons that are connected to each other. And together, they can do some amazing things, but actually, by itself, each neuron is very simple. It takes inputs from other neurons that come before it. If these inputs exceed some kind of threshold, the neuron becomes active and then sends an output to the neurons that come after it. So this is almost the most basic form of computation you can imagine, like a transistor. So since the very beginning, neuroscientists and engineers and physicists benefited from an interaction between each other to try to figure out how neurons and neural circuits worked. So some of the earliest insights into the electrical properties of neurons came from viewing them as an electrical device, just like a radio or a microphone. And in turn, observations for how biological neurons operate and how they're connected to each other inspired computer scientists to program artificial versions of the same neurons and link them up in their code. And this ushered in a new paradigm for computation, which is the neural network. And to get from these early neural networks to these fantastic systems that we have today took the work of many, many researchers over a long period of time. And so the 2024 Nobel Prize in Physics, awarded a few months ago, recognizes some of this work. It was awarded to John Hopfield and Geoffrey Hinton for foundational discoveries and inventions that enable machine learning with artificial neural networks. And actually, these discoveries also helped us better understand biological neural networks as well. So the press release goes into it a bit further. John Hopfield invented a network that uses a method for saving and recreating patterns. So in our brains, this is equivalent to storing and retrieving memories. When the Hopfield network is fed a distorted or incomplete image, it methodically works through the nodes and updates their values so the network's energy falls. Okay? Geoffrey Hinton used the Hopfield network as the foundation for a new network that uses a different method, the Boltzmann machine. This can learn to recognize characteristic elements in a given type of data, and actually, we'll see that it can generate new data based off of this. So this sounds maybe very mysterious right now, but hopefully by the end of the presentation, you'll at least get a feeling for what this means. Okay, so let's start with the Hopfield network. Like any other neural network, it's composed of neurons. And when it receives an input, certain neurons become active in the network. Now, in terms of, for the input, if for a biological neural network like those in our brains, the input might be an experience, like the one that we have here. The sound, the smells, the sights that we have. For a machine learning neural network, this input might be the training data set used to train the network. So I'll use these terms interchangeably. Okay, so which neurons are activated by this input? Well, that's determined by a certain rule. In biological neural networks, this rule is governed by the complex pathways of neural connectivity in the brain. So we don't really know this rule, and actually a big part of neuroscience is trying to figure out what this rule is. In artificial neural networks, well then the rule is determined by the programmer, the engineer who makes the network. So one such rule is that each neuron is responding to one pixel in the image. So if the pixel is dark, then the neuron becomes active. Instead, if the pixel is light, then the neuron remains inactive. An alternative rule is a bit more abstract. Maybe each neuron represents a certain concept that is possible. So if this concept is present in the image, then the neuron becomes active, and if it's not, then the neuron remains inactive. Either way, we say that this pattern of neural activity, kind of the state of the network in this neural network, it somehow represents or encodes this image. So this is the network's representation of this element of data. In the Hopfield network, there's a special rule in that any pair of active neurons grow a connection between them. And so you can see here, all the active neurons will gain a connection between them. And so then when you show maybe another piece of input to the network, there's different neurons that then become active, and connections between those active neurons are also growing. Okay, and if you take away all the inputs, then the network becomes inactive. But you notice that the connections that it has experienced remain. So in the Hopfield network, memories are stored in the connections between neurons. And this is actually the same as what we think happens in the brain. So in our brains, the memories are stored by these connections between the neurons. Okay, so that was that was storing these memories. Now, how do we retrieve these patterns of activity? So for this example, we'll return to this kind of concept version of our neurons. And imagine that someone asks you, oh, Edgar, I heard that you recently had this trip to Mount Fuji. It was so beautiful. Can you tell me about it? It said, yes, the mountain was beautiful. There was this wind coming off the lake, and I felt it. And there was something in the lake. It was really beautiful. What was it? Was it a bird? Well, what happens is that when you're kind of cued to remember something, then you start with this incomplete version of the memory. And through the connections that were learned before, the active neurons then activate the remaining neurons, and this completes the full memory, and you remember, okay, it was the boat in the scene that I had. And just as a technicality, the three neurons on the right, they are only connected to one active neuron, and so in this case, that's not enough to activate them, so they remain silent. Okay, so we can see that this Hopfield network can store and retrieve memories according to this rule, but Hopfield's perhaps his greatest contribution was not just designing this network, but actually creating this an abstract version of it, kind of a different interpretation. So in the Hopfield network or any other neural network, there can be many different states or patterns of activity that the network can take. Now, we can imagine arranging all of these different patterns of activity along a landscape, such that similar patterns of activity are located closer together on this landscape. And now what Hopfield showed was that the process of seeing an input, activating certain neurons, and building connections between them is mathematically equivalent to carving a well into this landscape that is centered at this pattern of activity. So that's for one pattern, and then it still happens for the other one as well. There's a different well carved, centered at the other one, and so on. Okay, and so during the process of retrieval, what happens is that it's as if you place a ball representing the state of the network at this incomplete pattern, which is close to one of your complete patterns, and the process of recovery is mathematically equivalent to the process of the ball rolling down the landscape, as if under the influence of gravity, reducing, let's say, its gravitational potential energy. Just go rolling down the hill, basically, and this completes the full memory. Okay, so what is the point of this? You know, we're perfectly able to understand the network without this whole landscape thing. Why do we bother this? It seems a bit extra. The true power of this interpretation comes when you kind of forget about the physical connections in the network, which are actually the physical things that happen in your network, and you treat this abstraction as the central object to be studied. And so this process has been used before in physics to kind of create breakthroughs. There's like the field in quantum mechanics and the metric in general relativity. And the idea is that you treat the abstract version and you kind of take it for real. So you can wonder, can we create richer landscapes with more complicated features? And instead of having the ball simply rolling downhill, can we have it explore all of these rich features of the landscape? So this is what is accomplished by the Boltzmann machine. So Geoff Hinton's Boltzmann machine extends upon the Hopfield network in 3 key ways. The first way is that he adds more neurons to the network, the ones in gray. And these neurons aren't activated by the inputs in the same rule, but instead they're connected to the other neurons, and they're activated through these connections. So this represents a common theme in machine learning, that simply more neurons improves the computational power of the network. So in the Hopfield network, the number of neurons and your computational capacity was limited by, let's say, the number of pixels in the image, or the number of concepts in your concept list. But by adding neurons in this way, you're freeing yourself from this restriction. Okay, and the second key difference is that in the Boltzmann machine, each memory only creates a small change in the memory landscape. And so the landscape isn't built kind of by individual memories, but it's built gradually as the network accumulates more and more images and the large amounts of data. And so now this final landscape, it doesn't represent individual memories, but it represents the common features across them, kind of the statistical distribution of the data that you've been presented. And then the third key difference in the Boltzmann machine is that there is a bit of an energy added to the network state, the ball, so that it naturally, kind of like a vibration, will move around the network. So in physics, this is equivalent to a temperature. But the idea is that it'll traverse around all the features of the network, and it might end up in a place corresponding to an image that is never seen before. But still, it produces this new data that is based off of all of the images that it has seen before, based on the landscape that was created by all these other images. So this is an early example of generative AI in which your network produces new data that was based off of training data. Okay, so now if we go back, we can understand that Hopfield created this network that saves and recreates these patterns of neural activity. And when it is fed a distorted or incomplete image, it methodically works through the nodes and updates their value so the network's energy falls. So the network's energy falling is the network kind of rolling down this memory landscape. And we can also kind of appreciate, at least, that Geoffrey Hinton created this Boltzmann machine, which can learn to recognize characteristic elements in a given type of data. And we can see it can even learn to generate new data based upon it. Of course, with any type of research, it doesn't happen in a vacuum. And so this work was influenced by a lot of other work that preceded it. Interestingly, in the Wikipedia article for the Hopfield network, some of these other contributors were named in the intro sentence. Recently, however, it has been changed to a more simplistic form. I'll just highlight a little bit of work done by Shunichi Amari. Admittedly, he is a colleague at my institute, Regan Center for Brain Science. So yeah, I'll just point out that many years before the Hopfield network, he created a network that has the same rules for building connections between neurons, the same rules for activating individual neurons, and also the same inspiration from neuroscience, citing work by Donald Hebb here. I would say that Hopfield's contribution was really popularizing this network, and by creating this energy landscape interpretation that allowed people to really, extend beyond it much more easily. Okay, so let's kind of leave the past where it is and come into the present now. And of course, today we have these generative AI machines that can do amazing things. And all of this was made possible indirectly by the work of Hopfield and Hinton. But in fact, more recently, it has been appreciated that the machine learning neural networks used in ChatGPT, the transformer architecture, as well as used in Stable Diffusion, the diffusion model, they're very much related to the Hopfield network and memory networks. So in fact, the Hopfield network and the Boltzmann machine, their concepts are very much influencing machine learning research today. And the same concepts are also influencing neuroscience research for how we store memories in our brain. And of course, there's many people working on this, but I'll just present some of my work in this direction, along with my collaborator, Taro Toyozumi. We store memories in an area of the brain called the hippocampus. And in particular, there's an area within the hippocampus called CA3. And CA3 contains neurons that are connected to each other in a way that very much resembles a Hopfield network. And people believe that it stores memories in a way that's similar to a Hopfield network. Now, the inputs that it receives, all the sensory information that we want to store as our memories, come from an area of the brain called the entorhinal cortex. And entorhinal cortex sends this information to CA3 actually through two different pathways. First, there's this orange pathway called the perforant path that goes directly to CA3, but before stopping there, it branches off and has a second path, the purple path, called the mossy fibers. So the question is, why do we need these two different input paths that are kind of conveying the same information? What is the computational purpose of this? Well, to kind of address this, we created a neural network model of this area where we modeled these different regions, including modeling CA3, the memory network, as a Hopfield network. And the experiences in our model that we want to store as memories are these images, each one of which is an example of a concept, either sneakers, trousers, or coats. And what we find is that these two pathways store memories. They carve different types of features into the memory landscape. So the orange pathway carves features that are a little bit broader, and the purple path, their features are a bit more narrow. In addition, when it stores more memories, well, similar memories are located more closely together in the orange pathway, and they're located a little bit more farther apart in the purple pathway. So yeah, by accumulating more and more of these memories, you can see that, well, in the purple pathway, you can always store and retrieve the exact memory that you put in. So all of these shoes are quite distinct. But in the orange pathway, all the shoes kind of merge, all of their wells merge, and all the trousers merge. So what you do is you build concepts of each of these categories of sneakers, trousers, and so on. And so this is showing that, these two pathways in the hippocampus can be storing the same information, but in two different ways. And these ways can be important for different tasks. For example, if you're walking along Tasmania and looking at ferns, and you see this new fern that you see a fern on the ground, and maybe you want to remember, is that exactly the fern that I saw before? So in that case, maybe it's more helpful to use the purple path, because that can really tell you, okay, it's this one and not that one. But maybe you're more you see this new fern, and it's I've never seen it before. Is it a fern, or is it the philo, the one that's related to the fern? Is it a lycophyte? I've never seen it before. So, like if you were to use, if you were to use the other pathway, then maybe, you wouldn't know where to put it. But in this one, in the orange one, you would be able to easily characterize, this is a fern and not a lycophyte. Perfect. Okay. And what my group is working on right now is expanding this idea of energy landscapes and memory in the brain to study diseases, and in particular to study seizures. So you can imagine two different regions of the brain that are storing different types of memories with these kinds of energy landscapes. Now, you can imagine that a seizure might be adding just a lot of internal energy to the network state, such that it really buzzes around a lot. Well, you can imagine that in the left network, the barriers between these memories kind of constrain the activity of this runaway activity, so that only a few neurons are active. But you can imagine that in the right landscape, because there are these barrier-free paths, then this runaway activity can be quickly amplified. And so maybe these kinds of networks are more susceptible to seizures. So this is something that we're testing right now. Well, I hope these examples kind of gave you a sense of the value and the promise of these Hopfield networks and the Boltzmann machine, and how they might carve a path into a bright future for AI, neuroscience, and maybe even medicine. And with that, I'd like to thank kind of the network of people that have helped me with my research, and thank you for your attention.
The presentations, it's very interesting. I want to ask about the architecture, the different types of arranging these neurons. Today you mentioned about Boltzmann machine and Hopfield network. They are a very great pioneering works, but I don't think they're used much in modern AI, let's say. Do you know why? And also, well, nowadays they usually use the feed-forward neural network, different types of ranging this neuron. Do you know why is that so? And the second thing is, well, I guess modern AI has some types of memory as well, but they're not used a lot of Boltzmann machines or Hopfield network. And so what make you think that our memory are based on Boltzmann machine or Hopfield network instead of this other feed-forward neural network? Sorry. Thank you.
Great, yeah. Thank you for the questions. For the first question, maybe the person saying next to you might give a better answer the other way. Yes, Ali, but I'll give an attempt at it. So these early networks, a long time ago, there weren't so many neurons that were available to them. They could simulate networks of maybe a few hundred. And so the learning rule that they have, the particular way that they carve things, they carve relatively broad wells into their system, just because there wasn't so many neurons, and you wanted to kind of capture the data. The modern machine learning architectures like the transformer architecture and these diffusion models, they can be interpreted as like an extreme version of Hopfield network where you're covering very, very sharp wells into the network. So you have, you can chisel away the landscape with a much finer chisel, but that's only possible because you have so many neurons now. You can simulate things with so many neurons. Whereas before, if you use such a small chisel and you only have so many neurons, then most of your network's space would be, you couldn't have any features there. So that's one maybe, I hope it's true for answer to the first question. The second question of, why do our brains match Hopfield networks and maybe not some of these modern machine learning architectures? I think honestly, the brain is probably very different from both of them. But because the Hopfield network is simpler, that's kind of our first approach to drawing some kind of comparison. But yeah, I think the way that the brain does things is much more complex as well.
Okay, I have actually two questions that are a little bit extreme, so I'm sorry. The first one is, what about non-neurotypical people way of interpret information inside of these analogies of the human brain? Because they are definitely different, and that's one source of question. The second question is about the suspicions of quantum phenomena happening in the brain and in the way the brain is working. I'm thinking about Penrose model of the quantum brain and others' suspicion of the brain being room temperature quantum computer, or at least partially.
Great. Thank you for these extreme questions. I think, it might be difficult for me to answer either of them. I mean, so kind of related to the last question. I think the brain it's so complex, and we're just trying to figure out, even how flies have memories, how rats have memories, and so there's probably many people that have a good understanding of what features of memory, let's say, are different between neurotypical and neurodivergent people. But I think at this level of detail, I don't think we know exactly, what is the distinction there. In terms of the quantum brain, I think that's also very difficult for me to say, because for me it's quite difficult to imagine that, basically a room temperature quantum supercomputer, it's difficult for me to imagine, so I would have to kind of look into that a bit more deeply.
Yes, so at least a bit of happenstance, because the suspicion is something hybrid with, I would say, a random event generator coming from the quantum phenomena. Most of the activity being classical computing.
I see.
Some additional spark of something that is much more complicated to modelize.
It's always possible. In my view, the brain is actually surprisingly robust. Like if you were to eliminate a neuron, or if I were to scoop out a piece of brain, I don't know if you guys have tried it before, but you can really scoop out a piece of brain, and often patients, they would have very little deficits, and so unless if it's in key locations. So to me, it just seems that the quantum perturbation, it would be kind of subtle. So but it is I would be very excited, coming from physics background, if quantum mechanics had anything to do with the way we think.
Okay.
Thank you.
Okay, sorry. So I'm a...
Oh.
Do we have a little bit of commentary on the first question before we move on to the second question?
A quick acknowledgement that
Wait. Talk into the microphone, otherwise the people on the live stream can't hear you.
Oh, people live stream.
So a quick acknowledgement. I actually used to do my postdoc with this at the same lab that Lewis is collaborating and reported. So back to the question of why did the industry kind of separate it from this more realistic architecture, is that money and scalability, but the downside is that a lot of the drawbacks of today's transformer architecture people are talking about directly link to the lack of the kind of feedback connections that Luis was talking about that are present in the original computational model of the brain and not the transformers. Now, I want to ask you, Luis, in the last 10 years, because I kind of left academia 10 years ago, have there been more computationally tractable ways of modeling these feedback connections that are basically missing in transformer architecture?
Do you mean in the machine learning context, or the neuroscience, or either?
Well, let's start from computational neuroscience and how much of it could be applied.
I see. Yeah.
For machine learning.
Thank you for that question. It's been, I guess, being more appreciated now that feedback connections are ubiquitous in the brain, even in areas like primary visual cortex that just thought were just, responded to light. There's actually a lot of modulation from feedback from higher up structures. And so, yeah, I think there is a lot more understanding of that and in incorporating that into models, and how that achieves more complex computations for one, but also kind of, does good network husbandry and kind of maybe keeps the networks in good operating condition, like balancing excitation, inhibition, these things. Leave it at that for now. Yeah. Thank you, Ali.
Okay, I'm a practitioner of deep learning myself. I work mostly with natural language processing. So like the current way that we're modeling with natural languages, we have the idea that there is a specific set, at a specific time, there's a specific set of outputs. So we have a species present, so to speak. I imagine with the brain working at it is with like biological models, they don't have a synchronized clock, right? So what I'm trying to ask is, have you had experience where some features are time encoded? So like patterns of patterns across time encoding certain phenomena, as compared to like a single set of embeddings, single set of values encoding every single phenomena that are possibly representable.
Yes, thank you for this question. The answer is yes, and maybe I'll give kind of a brief answer, just because it's just, these sequences, these temporal sequences are also ubiquitously present throughout the brain. So these kinds of, process where the network falls into the well and stays there, that is a vast simplification. And it actually, some people think that the primary purpose of the brain is to remember sequences, not distinct memories, which can help in things like prediction, that sort of thing. So maybe I'll just leave it at that. Yeah, thank you.
I think we got time for one more.
Thanks. I am not a specialist in artificial intelligence or anything, and I have a pretty basic question. So the first half of the talk seemed to be fairly abstract in terms of how these networks are set up, and then you're looking at what's going on in the brain. I was just wondering how you actually empirically test what is going on in the brain and how to match that up to these much more abstract, networks.
That is a great question, and I think it's a big problem in neuroscience today that there's a lot of ideas out there, and they kind of seem to float. I guess one advisor once told me that like a good theory can be a theory that is like perishable, like it can be killed, I guess, it can be tested and then go off to the theory graveyard. Otherwise, there's just all these theories that are floating about. So, for example, in the work with in hippocampal encodings, we first simulated the network based off of the architecture, and we found these types of complementary encoding, the orange and purple encodings. And then from that, we identified kind of a neural signature based off it. And so then we tried to look for the signature in data, and we found we found this signature in the region that we were expecting. So that doesn't, fully prove that it's there, but it does support it. So I think it's important to test with experiments in these ways.
Okay. Lewis, thank you so much. Can we get a big hand for Lewis, please? Both speakers, before you go, I got to get a photo with you, so don't disappear on me, okay? Lewis is going to be hanging around after he's got that thing off his head, and you can still probably ask him questions. We do have to hustle everybody out by 10:30, but we got a little bit of housekeeping to do first. So let me just do this, and let's see if this works for once. Let's see. Okay, that's for the background of my... Okay. All right, so we... let's see, does this work? Boom, there we go. So in the, I think it was the 70s, hold on, there's going to be a clunk. Oh, nice. Why is this so short? Hold on. Oh, there, okay, there's the thingy. Whoop, where's my roadie? Yeah, so we're about the same height, right? This is going to be okay for you. So, there was a, George Carlin did his bit about the 7 words you can't say on television. This is actually a photo of him being arrested for doing that bit. Those words were shit, piss, fuck, cunt, cocksucker, motherfucker, tits. Actually, it was a mixture of those words. He would revise them over the years, but he said, people complained that motherfucker was kind of just a duplicate of fuck, but he said you can't leave it out because the rhythm doesn't work. But this is all as an introduction to something the American Andrew wants to talk about. So, did you want to come up here? I could go to the next slide if you're embarrassed to be. He doesn't want to stand next to the words. He's going to read you all of these.
Yeah, I hope you're all settled in. We're going to read all 109 of these. Yes, thank you, Canadian Andrew.
Soon to be American Andrew.
Soon to be American Andrew, possibly. Yes, so thanks. I'm also Andrew. I am from America. I grew up close to Washington, D.C. In my younger years, I was an enthusiastic student of governance, politics, public policy. When I was in high school, I actually interned at the State Department. And now I'm your speaker recruiter here at Nerd Night. And it's been a difficult month or year or whatever the last 6 weeks have been. And I think we all know that. I think we all have some awareness of what's been going on. And this obviously isn't the place to get into everything that's going on. It's been difficult to watch. It's been much more difficult for a lot of people who are more directly affected. As many of you might know, this is old news by now. Pretty much all U.S. government agencies have been directed to find a way to essentially eliminate concepts that the incoming administration feels should not exist despite existing, despite being a part of reality. This is an example at the NSF, the National Science Foundation, who will now essentially flag any grant requests that come in containing any of these words, which I understand are quite difficult to read on here, but I think you can guess what's on this list. A few that... Yes, words like woman or female are on this list, along with things like diversity, support, trans, items like this. Some items that aren't on this list but are on the list of other agencies are things like climate change. So all I'm here to say tonight is, if you would like to give a talk on any of these subjects, please come talk to me. Do we have anything else?
Yeah, I was going to say, this is fine, however. Do you have something to say to wrap this up, or?
Just don't forget that we're in Roppongi next month.
Yeah. So yeah, that's basically it. But oh, excuse me, how did he get there? Yeah. So yeah. The preceding they did not. Yeah. Yeah, I'm. That's just me. That was solo. Yeah. So that's it. Yeah. But Bruce, American Andrew is right there. If you want to do a talk about any of those words or multiple of those words, please come talk to us and we will find a way to fit you in. Having said that, yeah, we're going to start packing up. You can hang around for a little bit. We have to be out on the street by 10:30, so we might start hustling you out the door in about 20 minutes as we finish packing up, just so that we're not stuck here. But in the meantime, you're welcome to stay here. 2 speakers, please come over here so I can take your photos. Don Warren, please stick around. I want you to photobomb that photo. And yeah, thank you everyone for coming. Big hand for our 2 speakers one more time. And hopefully we'll see you next month in Roppongi.